Hello, my name is Nathan Novy, and I'm a graduate student in the Raman and Romero labs at the University of Wisconsin-Madison. And today I will be presenting my research related to phage therapy. Over the last roughly 100 years, we've been fortunate to have antibiotics, which could be life-saving medicines against bacterial infections. However, the era of antibiotics is ending. Antibiotic resistance is responsible for the conclusion of this class of drugs because the process reduced the effectiveness of many currently available antibiotics. Antibiotic resistance occurs because of occasional mutations in bacteria, allowing the bugs to slightly overcome the effects of antibacterial drugs. Bacteria with these beneficial mutations survive and replace the bacteria that cannot resist the effects of antibiotics. This process repeats, slowly allowing bacterial populations to get better at surviving in the presence of antibiotics and is an unavoidable cycle when antibiotic use is widespread. A promising alternative to antibiotics is bacteriophage therapy. Bacteriophages, or phages, are viruses, and the term bacteriophage in Greek essentially means bacteria eater. This means that unlike the viruses that cause COVID-19 and the common cold, phages can only infect bacterial cells and are harmless to humans. In phage therapy, bacteriophages are administered to a patient to target and kill harmful bacteria in the body, producing the same effect as antibiotics. Phages are actually the most abundant organism on the planet, and they are wherever bacteria are, which is everywhere. The diversity of phages is one reason why phage therapy is such a promising alternative to antibiotics. However, this diversity also creates a challenge for researchers because scientists need to be sure that they use the correct phages when these viruses are applied as therapies. So what do the right phages look like? Phages used as therapies need to target harmful bacterial species and avoid the many species of bacteria that are important for your health, like the bacteria that live in your stomach and help you break down food. However, isolating natural phages that are both safe and effective as therapies is very challenging and time-consuming. In my research, I consider an alternative approach. What if, instead of finding phages that are safe and effective, scientists could engineer these phages in the lab? This is a 3D printed model of a bacteriophage developed by the members of the Raman lab. As you can see, phages look a lot like spiders. The core components of a phage are a head that stores the DNA and these arms that allow the phage to grab onto bacteria and infect its targets. In my research, I am modifying the DNA stored in the heads of these phages to change the structure of these phage arms and engineer the phages to target specific bacteria. However, there are literally trillions of different ways I could engineer this phage arm, and I don't have enough time to test all of these options. So instead, I apply machine learning to help me predict which phage arm variations are the most probable candidates for being able to target harmful bacteria and avoid good bacteria. To test my method of engineering these phages, I have designed thousands of variations of this phage arm that our machine learning algorithms predict will be able to target different strains of bacteria specifically, and I'm currently testing these engineered phages in the lab. My hope is that this research will help advance the application of phages as therapies, and that one day this research will help save lives when antibiotics no longer can.